Hey everyone, let's do transcription and translation now. So in the previous DNA video, we saw how DNA gets replicated during interface of mitosis and meiosis, and it also happens the same way in binary fusion. But now we're going to talk about transcription and translation. So to be clear, DNA replication, we should all know, happens in the nucleus of a cell. Now, this would be the nucleus specifically of eukaryotes, right? Cells of plants and animals. But where would DNA replication happen for a bacteria? Well, for prokaryotes, that would happen in the cytoplasm because they don't have a nucleus, right? Okay, that's important to note because when we do transcription, notice that you're still in the nucleus, right? So transcription for eukaryotes is also happening in the nucleus. But because bacteria don't have a nucleus, transcription for them is also happening in the cytoplasm. All right, so make sure that you make a note of that. So what is transcription? That's the conversion of DNA into a different nucleic acid language called RNA. They are both serving as instructions for building protein. The stored instruction is in the DNA, but the instructions that get to come out of the nucleus so that you can build a protein, that's RNA. So let's take a look at both transcription and translation and how they're related. Now, before we move on from this slide, you should know that the copying of DNA by eukaryotes, plants or animals, is only happening in the cells that are doing mitosis or meiosis. So if you are a nerve cell or a muscle cell, you're not copying your DNA. You need to remember that from our discussions of mitosis and those checkpoints. Remember that you need growth factors to initiate the copying of DNA. And if you don't listen to those growth factors because you don't have receptors, then you're not copying your DNA. But transcription and translation, that's happening in all cells, not just the cells that are doing mitosis and meiosis. Transcription, translation make protein, and all cells need protein. So all cells are doing transcription and translation. All right, so here's an example of DNA, and you can see that there's 1, 2, 3 genes that are highlighted. And what we're doing is we're going to take a small section of gene 3, and we're looking at it here now. And you can see the DNA strand contains A's, C's, G's, C, A's. And then take a look at transcription. Transcription shows us how we are rewriting the language from DNA to RNA. Notice that the A's are no longer bonded with T's. Now they are bonded with U's. Notice that the C's and G's are still bonded to each other, the G's and C's, right? But whenever there's an A, instead of having a T, now it's a U. All right, we're going to talk about the differences between DNA and RNA in a second. And then further down, notice that in the process of translation, we're taking the nucleotides of RNA, 3 at a time, and we're using that to call it a codon, and that's going to allow us to put in a very specific amino acid. Here's another 3 nucleotides called a codon. They are different from the first 3. That gives you a different amino acid. Here's another one, another codon, and because it's different from the other 2, it gives you a different amino acid. Here's a 4th codon, And notice that that is the same as the first one, U. So it gives you the same amino acid. We're going to work our way to teasing out those details. First, what's the difference between DNA and RNA? The bases of DNA include A, T, C, and G. But in RNA, they are A, U, C, G. Notice that the T's and the U's are the difference between the two. There's some other differences, but that's a really big difference. There are no T's in RNA. There are no U's in DNA. And that's how you know if you are looking at DNA or RNA. For example, if you see this, you should know that's DNA. And you should know it's DNA because of the presence of that guy. On the other hand, if you see this, then you should know that's RNA because of the presence of that guy. Okay, that's the difference. The sugars on DNA are called deoxyribose. The sugars of RNA are called ribose. And the differences are basically that by saying deoxy, that means you're missing an oxygen. But in ribose, you have the oxygen. So we can say that deoxyribose is a little incomplete because it's missing an oxygen, whereas ribose is complete. It has that oxygen. Lastly, DNA is double-stranded. RNA is single-stranded. You might remember that DNA is made up of a DNA double helix. But RNA is just one strand. All right. When we transcribe, we're going to use an enzyme very similar to DNA polymerase that you saw in the DNA replication lecture. This one's called RNA polymerase. DNA polymerase makes DNA, RNA polymerase makes RNA. And the RNA polymerase also does more things than DNA polymerase. DNA polymerase, for example, cannot unzip the DNA. It can't separate the DNA. You need a DNA helicase for that to open it up. RNA polymerase can do both. It can unzip the DNA, separate the two strands, and then it begins reading the DNA to create RNA. We're going to summarize that in a second. Focus your attention here, and you'll see RNA polymerase has unzipped the DNA. It separated the two DNA strands. That's the like the fork that you saw in DNA replication. And then notice that it's reading the bottom strand. And it's only reading one strand. It's not doing both. Wherever it sees a G, it puts down a C. If it sees a T, it puts down an A. You're seeing it here. When it sees an A, it puts down what? A U. And that's how you know it's making RNA, not DNA. Okay. Also, notice that it's only one RNA strand that's being made, not two. All right, let's look at the rules for transcription are super quick and easy. They are read DNA 3 to 5. That should look familiar. That's the same rule for copying DNA. So whenever you read DNA, you always have to read it 3 to 5. Whenever you're copying DNA, whenever you're transcribing DNA, it's always 3 to 5. And also, when you create RNA, new RNA is made 5 to 3, just like when you make new DNA. New RNA is also 5 to 3. And so those are very much the same rules that you saw in DNA replication. So that makes it easy to remember. And finally, there's only one enzyme here. It's RNA polymerase. And that guy's job is easy, makes new RNA from DNA. One enzyme. All right, so this should be simpler than copying DNA because copying DNA has more rules and it has more enzymes. This one has one less rule and it's got only one enzyme. Okay, so here's something interesting also. Let's say that we had a DNA strand here. I'm going to call it DNA. And I'll make my, I might color code it here so that we can tell it apart from RNA. If you are RNA polymerase, let's maybe make that guy in blue. If you're RNA polymerase, how do you know where a gene begins and where it ends? Let's say that the gene is from here to here. If you're RNA polymerase, how do you know that the gene begins there and that it ends on the other green hash mark? Here's why I ask that. You have to imagine yourself as RNA polymerase. And whenever you unzip DNA, and here I'm only showing one DNA strand, but when you read that DNA strand, you're going to be reading CCGTA, TTC, GGCA, ACG, TC, TT, GG, CA, TTA, GGC, TA, TTA, GC. It's all a bunch of gibberish. It's just a bunch of A, C, T, and G in random orders, random combinations. How do you know where to begin transcription? How do you know you found the gene? And how do you know when to end? Because it's all a bunch of gibberish of 4 letters. So how do you know? And the answer is we know because at the very beginning, before the gene, something interesting happens. And after the gene, also something interesting happens in that gibberish. Here's what we learned. Whenever the RNA polymerase gets close to the beginning of the gene, in this region right here of the DNA, there's a very specific sequence. It might be something like AAA, TT, AAA, TT, AAA, TT, AAA, TT, AAA, TT, repeated hundreds of times. AAA, TT, hundreds of times. No C's, no G's, and always AAA, TT. And now that seems to be like something that's calling your attention. It's indicating to you that the gene is coming up. We call it a promoter sequence. So you're reading AAATT. Remember, you are RNA polymerase. And you're reading AAATT, AAATT. And then all of a sudden it goes GCATC. Okay, now you're at the gene. You finally got to the gene because you broke that repeating AAATT sequence. Similarly, how do you know when the gene has ended? Well, at the back end, if you're thinking you're going to find another AAATT, you have the right idea. but it wouldn't be that sequence, because remember, that sequence starts transcription. So now it's going to be a different sequence, and that one could be, for example, I don't know, I'm just going to make up something. C, 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 G, 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 and it's repeated like a couple hundred times. And that's going to be called a terminator sequence. All right, so in summary, during transcription, where are the genes located? They're located between what? They're located between a promoter and a terminator sequence. That's how we know where the gene is. That's how RNA polymerase knows where to begin transcription. It'll actually land right here, open up the DNA, read it, or read it as it's continuously opening DNA. And then it'll begin transcription. It'll start making the new RNA strand. As it's making its way across the gene. And how does it know to stop? It'll get to this terminator sequence there. And that's when it releases the RNA. I'm going to show you a video on this. Okay, so let's pretend that we made this new RNA. Okay, I'm going to just kind of make it bigger. And I'm going to give it the title of new RNA. And what makes it RNA is, again, it's going to have a bunch of U's in there instead of T's. But once we made the new RNA, I like to call it raw RNA because it's not ready. In eukaryotes, that raw RNA needs to be processed. And we do the processing in 3 steps. First, you should know that the RNA comes in segments. Like, for example, something like this. I just made up some cuts in there, segments. And at the very beginning, I'm going to call this, there are exons and there are introns. I'll get back to what these are, okay? And I'm going to say that this is exon number 1. This is intron number 1. This is exon number 2. This is intron number 2. And I think you can see the pattern. This is exon number 3. This is intron number 3. And this is exon number 4. And what we're going to do is we're going to get rid of the introns. And then we're going to piece together the exons. So that exon number 1 is about that length. Exon number 2 is a little shorter. It's about that length. Exon number 3 is a long one. And then there's an exon number 4 that's about the same length as exon number 1. All right, so these are the exons 1 through 4 linked with the introns removed. All right, what are exons? What are introns? Exons stand for expression sequences. We need them to express, to make a protein. Introns, on the other hand, are intervening sequences. They are in the way. They're intervening. We got to get rid of them. All right, and once you do that, you have done a very big step in what is called processing of RNA. There's 2 more things to do. We're going to add a whole bunch of A's at the back end. And we're going to put a guanine up at the front. We call this a G cap. So the 3 steps are, I'm going to write here, in 3 steps. All right, step 1, remove introns to connect exons. 2, add a GCAP. It's made out of guanine. 3, add a poly A tail. That's the poly A tail. There's the GCAP. And when you do that, you will have the following. Well, let's make it better looking. You'll have a guanine cap. You'll have a bunch of A's here. A whole bunch of them. And now you have something called messenger RNA. Remember, all of this is happening in the nucleus of a eukaryote or in the cytoplasm of a bacteria. If it's happening in the nucleus of a eukaryote, this messenger RNA is now able to leave the nucleus. So the first thing you have to do is transcription. That's what we did over here. And then you have to do RNA processing by doing those steps. Now you have messenger RNA. Now you can leave the nucleus. This is what we just did. This is DNA, and we did transcription. And notice that we then have added a cap, a tail, and removed introns. I don't like the order shown here, but doesn't matter. You know the 3 steps. And when you're done, you will have created messenger RNA, and now you can leave the nucleus. You can see that happening there. Okay, let's do a little practicing of this. In other words, let's work our way towards translation. Here's what we've done at this point. We copied our DNA. We transcribed our DNA. RNA polymerase has found the promoter sequence, read the DNA, transcribed it into making raw RNA, and then stopped when it reached the terminator sequence. We removed our introns, right? Those have to be removed. We put together exons, and that shortened the RNA piece. We added the guanine cap. Here I'm showing it to you a little more detailed. We added a poly A tail as well. I'm just cleaning it up. And now in black, down here at the bottom, here, oops, here, I'm showing you the RNA, and I want for us to read it 3 nucleotides at a time. And if we do that, we're now doing translation. We're reading 3 nucleotides at a time to decide what amino acid to bring in. So we're about to do translation. We've already done transcription. So here's the way translation goes. We're going to read 3 nucleotides at a time. And as we read, we decide from those 3 nucleotides, what is the appropriate amino acid to bring in. We have a choice of 1 out of 20 amino acids. we will know which of the 20 amino acids to bring in based on those 3 nucleotides that we are reading at a time. Remember, this is a codon, right? It's made up of 3 nucleotides. So we could say this is codon 1. The next one would be codon 2. The next one would be codon 3, and so on. We have 4, 5, 6, 7, 8 codons. So I'll clean this up a little bit. All right, so we have codon 1 through 8. I hope that made sense. Let's take a look at the codons. When we translate, we also have to start somewhere. It turns out that we're not necessarily going to start at codon 1. We're going to scan and we're going to look for a very specific codon, and that codon is called the start codon. And we all need to know what the start codon is. It's a very, very common start codon. It's the start codon everyone uses. Bacteria, eukaryotes, we all use the same start codon, and we need to find it. And then we need to find a stop codon. So let's practice doing that. I'm going to give you this table on exam 3, and I'm going to give you a short sequence like this one in black that has all the codons. And I want you to read your codon, find it here in this table, and then tell me what amino acid you need. So remember, we need to find the start codon. Have you found it? I think you have. It's in green, very obvious. Have you found the stop codons? There's 3 of them. I think you have. That's one, and you can see the other 2 to the left of that guy. So what's the start codon? AUG. Let's find it. So we're going to scan our RNA, and when we find the start codon, and I think we found it right here, we're going to label that the start codon. And now we're going to read from there, okay? But that start codon has a very specific amino acid. What's the amino acid? The amino acids are abbreviated. This amino acid is methionine. So I'm going to write MET right here for methionine. Again, it's right there, MET. If it's not obvious enough, we can blacken it. It's MET right there. Okay, let's look at the next codon. The next codon is UGG. Remember, we found the start codon. Now we're going to start reading to the right of that. UGG. Let's find UGG. Okay, where the first base, remember it was UGG. So the first thing we need is the U, right? That's the first one. And if we look at the first base, here's U. The second one we need is G. So let's look at the second base. That's over here. So if we bring those together, we're going to be in this box right here. And then the third one we need is G, and that's over here, right? That's the third base. So here it is, UGG, and what's the amino acid? TRP. So let's go back and let's put TRP there. That's tryptophan. These are the amino acids. and we are reading RNA and converting it to amino acids to create protein. Okay, what's the next codon? Looks like it's AAG. So let's find AAG. Okay, AAG. Okay, we go to the A's, we go to the G's, and find our box, and then, oh, I'm sorry, AAG. So we're actually AAG, so we're in this box now, right? Here's the A, the second A. So first A, second A, and now we go to the G, AAG. Looks like we need lysine, LYS. Let's put that in here. All right, and then we connect them. And I think you remember we connect them using dehydration synthesis. That was a while ago. That's how you create long chains of polymers. A protein is a polymer. And we have to use dehydration synthesis to build a polymer. Okay, the next one over is ACG. ACG. Let me write that down. ACG. All right, we go to the A, we go to the C, and we find that we're in this box, and here's the G. The amino acid is threonine. Come back here, THR. And again, dehydration synthesis to connect them. The next one is GAU. GAU. Okay, so we go to the G, we go to the A, we're in this box. Here's GAU, asparagine. So now we need ASP as the amino acid. We link those using dehydration synthesis, and we're down now to codon 8, UAA. All right, maybe we need to clean this up a little bit. Okay, we need UAA. So UA, we're in this box, UAA, and that tells us to stop. Notice there's no amino acid. Stop codons have no amino acids. That's how you know to stop, because they don't have amino acids. So we stop. Okay. How many total codons did we actually read? Did we read all 8 of them, or did we read some of them? It was only some of them. In other words, we did not read that one or that one. We can only read after the start, from the start codon over. So we read this one. That's 1, 2, 3, 4, 5. We read 6 codons. Not 8. Because we have to start at the start codon. Also, how many amino acids did we actually put down? 1, 2, 3, 4, 5. We did not put down 6 amino acids. Why not? Because the 6th amino acid is a what? The 6th codon is a stop codon, and stops have no amino acids. So if you had read, for example, 10 codons, you should know how many amino acids that would have represented. The answer would have been 9 amino acids. Again, because the last codon is a stop and it has no amino acid. Okay, let's do this now. use red to make sure we highlight this. When we had 6 codons, how many total nucleotides was that? Well, a codon is made up of how many nucleotides? 3, right? 1, 2, 3. Nucleotides. So if you have 6 codons and each one has 3, that's 18 nucleotides. And if you had 10 codons, the total nucleotides would have been 30. All right. You might need to go back and review that. So I'll give you this table on exam 3. Nobody memorizes this. You'll need the table. I'll give you RNA, and then you use this table the same way I just showed you to tell me what the new amino acids are, and then be prepared to answer these kinds of questions. Okay, let's go back to this translation step. Remember, translation happened once we had our RNA. Translation happened when we started here. Everything we did down below that, looking at AUG, reading the table, that was all translation. Transcription was converting DNA to RNA. Translation is reading the RNA to make an amino acid chain. So I want to talk about the types of RNA. There's 3 types. We'll stay in red. Number 1, we have messenger RNA. That's the one that left the nucleus. It's the one that has the poly A tail. It's the one that has the guanine cap. It's the one that's made up of exons linked together. This one's important because it carries the codons. And the codons are also known as triplet codons. That should help because they are made up of 3 nucleotides, which you already knew. But maybe calling them triplet codons will remind you of that. Otherwise, you can just call them codons. All right, the second RNA is really important. This one is called tRNA. Remember that the M stands for messenger RNA. The tRNA stands for transfer RNA. There were 3 RNAs that were involved in translation. We knew one of them. That's the one we were reading because it had the codons. The transfer RNA was the one that was bringing in the amino acids. This one's important because it carries something called anticodons, which is a new term. You haven't heard about that yet. I'm going to show you that in a second. And brings in the new amino acid. We're going to call it the correct amino acid. So it has anticodons, and it also has the amino acid. It's got 2 things. The anticodons are meant to read codons. They lock in together. And there's number 3, ribosomal RNA. This is basically the ribosome's RNA. And I think we're going to show it to you, but I'm not going to ask you anything about that on exam 3. I want you to know these two. All right, so not on exam 3. But I'll show you what it is. All right, so here is tRNA. This is tRNA. Notice it's one strand folded up all weird. Hydrogen bonds hold it together. Down here is an anticodon. And over here, we simplify the cartoon. And here's what we mean by anticodon. If the messenger RNA had AUG, the start codon, remember that's the codon, the anticodon would have been UAC. That's the anticodon. UAC. And that would bind to the codon on the messenger RNA. And then it would bring in the right amino acid, and I think you remember that might have been methionine. You can go back and take a look at that, MET. And we can go back. There's MET. If the next codon on the RNA was UGG, what would the anticodon be for that? ACC. And what would have been the amino acid in that case? TRP. Over here on the transfer RNA would be TRP. So I just want you to know that for every codon, there's an anticodon, and that anticodon is on the tRNA, and that tRNA always carries the correct amino acid. So that would be two different tRNAs, one that finds AUG, the one modeled here, and there'd be another tRNA that has the anticodon ACC and carries the correct amino acid, TRP. And on and on and on it would go until you build your amino acid. These two would be linked, and then we'd start linking the other ones. So it looks kind of like this. It's a big, messy ribosome made up of a large subunit in blue, a small subunit in a light beige. It has its own ribosomal RNA in here. In fact, the ribosomal RNA is a large component of the ribosome, so it's all over the place. A ribosome is actually a lot of RNA. Here, we've drawn it in a cartoon form, and we've shown you that it has two important pockets. And then over here is the way you want to imagine it. This is actually translation happening. All this mess we looked at here is happening in here. So there's your messenger RNA that has the codons. There's your transfer RNA that has the anticodons here. And then the ribosome basically serves to house them all, hold it all together. And then you can see the amino acid chain here being made, and you can see the new amino acid that's about to be added to that growing chain. Here's a little bit more of a stepwise fashion. First thing that happens is the small bun, the small subunit, shows up with the very first tRNA that is looking for the start codon. You might recognize it. You could see it right there. And then it brings in the correct anticodon with the correct amino acid. And what this small bun, the small subunit, and the tRNA are doing is they're scanning, they're looking for the start codon. And when they find it, they lock it in. So they've locked it in right there. And once they do that, that brings in now the large bun, the large subunit. And now you're ready to bring in the next tRNA. So we can begin up here, find the next codon, bring in the correct anticodon with the correct amino acid, bring it in, and then notice that the chain here, made up of 1, 2, 3, 4 amino acids, notice that that's going to be added to the 5th amino acid there. And you're looking at that step there. That's dehydration synthesis. And when they link them, you get rid of the tRNA that gave up the long chain. This tRNA is going to go back into the cytoplasm to look for another amino acid to add it here so that it can come back and play again because it gave up its amino acid. And then the process just repeats itself. And it's very fast. All right, so let's watch a video where we bring in together everything, transcription and translation.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, good. You might want to go back and review this lecture. In the third part, we're going to look at what happens when we don't copy DNA correctly, or what happens when we make mistakes. We'll talk about mutations. All right, we'll stop here.